In the last few videos, we looked at metabolomics methods, strategies for evaluating collections of small molecules from different types of samples, such as treatments of diseased samples versus controls, samples that were not diseased. Here, what we're going to do is extend this coverage of omics to include proteomics, the evaluation of not just one protein from a particular organism or cell, but instead a collection of multiple proteins. And much like the evaluation of small molecules from the perspective of metabolomics gave a much more comprehensive view of the overall chemical impacts of treatments on a cell, likewise, proteomics will give us a much more comprehensive perspective than looking at just one protein from a cell. In the next few videos, we're going to focus on some of the common strategies for proteomics analysis. In this particular video, what we are going to look at is proteomics that relies upon 2D gel electrophoresis coupled with mass spectrometry to enable us to detect the differences in the proteins between treatment and control samples, such as the difference in the protein profile between diseased versus healthy samples. As a general overview, which we'll get into more details here in just a few minutes, what happens during this process of proteomics with 2D gel electrophoresis and mass spectrometry is that first, samples of proteins from treatment and control are subjected to 2D gel electrophoresis. So the experimenter run 2D gels using treatment samples and 2D gels running control samples. Then those two gel types are compared for the treatment versus the control, sometimes by superimposing those using software. So for example, here in the middle of the screen is a representative image that shows the superposition of two-dimensional gels, one corresponding to a treatment and the other corresponding to the control. So in the case of the superimposed gel that's been evaluated using specialized software for this, what the orange dots represent are proteins that are only present or much stronger in the treatment. Blue spots, such as here, and elsewhere, represent proteins that are only present or much stronger in the control sample. And then the black spots correspond to proteins that are present in both the treatment and the control sample. And so the experimenter can then pinpoint which proteins are most evident in the treatment relative to the control. They can cut those particular spots, those particular signals out of the gel, and then use mass spectrometry to evaluate that particular protein in an effort to identify that protein and pinpoint differences between the treatment and control samples at the level of the protein. So to get into more details on how this works, the first thing we need to look at is the 2D gel electrophoresis step. In 2D gel electrophoresis, the separation of proteins from mixture typically happens in 2 dimensions. That's why we call it 2D. And those 2 dimensions are typically based upon isoelectric focusing in the first dimension. In isoelectric focusing, we are separating the proteins based on their different pI values. Remember that pI is the pH at which the protein has a 0 net charge. So isoelectric focusing is generally the first dimension in which the proteins are separated. The second dimension separates them based on molecular weight, much like a typical SDS page gel would separate proteins, much like you learned about in your biochemistry course where the proteins are denatured and then separated based on molecular weight. So as a result, what we end up with is a 2-dimensional plot of our proteins where in one dimension, we've separated based on the pI value through that isoelectric focusing, and in the other dimension, we separate based on molecular weight. And so as a result, we get a much better separation of proteins than would be possible by doing just one of these techniques alone. So what we would do from a practical standpoint is we would run multiple gels. We would run gels corresponding to our treatment samples, such as samples of diseased tissue from which the proteins had been extracted. We would also run gels of the control samples using the same conditions. In these gels, for the isoelectric focusing step, the way that this will work is that a pH gradient is applied to the gel along with an electric potential. Much like when running an STS-PAGE gel, there is an anode and a cathode, meaning that one end of the gel is positive, the other end of the gel is negative. And what's going to happen is that within that pH gradient, at all pH values other than the isoelectric point, the point at which the net charge is zero, the proteins will be charged and hence they will migrate toward the oppositely charged pole. If they're positively charged, they will be pulled toward the negative end of the gel. If they are negatively charged, they will be pulled toward the positive end of the gel, since opposite charges attract. On the other hand, once the protein migrates to its isoelectric point, the point where it has no charge, it will remain stuck stationarily there because it has no charge and hence will not be pulled any further toward the positive or negative direction. So hence, the proteins will be scattered across the gel in that first dimension according to their isoelectric points. So in both the treatments and the controls in the first dimension of isoelectric focusing, what's going to happen in this isoelectric focusing step is that the gel would be set up so that in both of these cases, for the treatments and the controls, since we're running an identical gel for each, but just looking for the proteins that differ between them, there would be a pH gradient across that gel. So within the gel matrix, the pH could vary throughout the gel across either a wide range or a narrow range. So in this particular gel example, I'm going to show the pH varying from a pH of 3 to a pH of 10. That's a pretty wide variation in pHs as we think about biological systems and the typical physiological pH being 7. So we've spanned a wide range around that. And at one end of this gel, we would have our positively charged pole. At the other end of the gel, a negatively charged pole. That would be true for both the treatment and the control. We're trying to run these as identically as possible. And then across this pH range, once we have loaded our proteins, if we have a protein that say that protein has a PI value for this particular protein, which we'll call protein X, has a PI value of 5.5. And keeping in mind that the PI value is equal to the pH where the net charge on that protein is 0. That is the pH then at which that protein will cease moving in the gel. At pHs that are greater than 5.5, this particular protein would have a negative formal charge because the pH is greater than the PI, meaning there's an excess of base, an excess of ability to take on a negative formal charge. And hence, at that pH range of greater than 5.5, the protein would be migrating toward that positive pole. On the other hand, when it is at the pH range within the gel that is less than 5.5, that protein is going to have a positive formal charge, and hence it would move toward the negative pole. And so as a result of this gel matrix being embedded with a gradient of pHs, what's going to happen for this protein that has a PI of 5.5 is that once it reaches the position in the gel, which we'll say is right here, is the pH of 5.5, it is going to remain right there. And so in this first dimension, the proteins would scatter across the full range based on their PI values. After that step had completed for both the treatment and the control, then what would happen next in the next step is that the proteins would be separated based on their molecular weight, going in the other dimension via a traditional STS-PAGE type gel method. And so after the isoelectric focusing, each of these gels would be taken out. It would be then placed into a gel apparatus for separating based on molecular weight, going in the other direction, the second dimension. So this dimension here, where we're separating based on molecular weight. And with the molecular weight separation, the lower molecular weight proteins will travel faster through the gel matrix than proteins with a higher molecular weight. And so what we would expect when they start traveling down the gel here, based on molecular weight, is that at the bottom of the gel, we would have the lower molecular weight proteins. Closer to the top of the gel, the higher molecular weight proteins. And this would be true for both the treatments and the controls, is that in that second dimension, they're separated by molecular weight. And so then once both of these gels, presumably with several replicates of treatment and several replicates of control, had been generated, what would happen then is that the gels would generally be stained using some sort of typical staining technique that you quite possibly have talked about in some of your other biological chemistry classes. So some of those staining methods would include very commonly the Kumasi stain, which if you have used the Kumasi stain, it causes the spots to turn out as kind of a purple color. Other typical stains include silver is a commonly used stain. There is also fluorescent stains that can be used to detect proteins, and things like autoradiography as well, depending upon exactly what proteins you are looking at in your treatment versus your control. So then at that point, once the samples have been stained, you would see, for example, in the case of a Kumasi stain, that your gel would look something like this, or gels would look something like this. So the gels, as a result of this 2D separation, would turn out looking something like this, where we've separated in one dimension based on isoelectric focusing, where as we go to the right, we would anticipate that the proteins over here had a larger pi value than the ones over here, and then separated in the second dimension, going down, as I'm showing with my red arrow, we would be going from the highest molecular weight proteins to the lowest molecular weight. So we can gain an idea about the molecular weight of each individual spot, where each individual spot represents a protein, and its pi point for our treatment compared to our control. Now, given the large number of spots here and the large number of different intensities of spots, it's generally not particularly possible to compare these two gels globally without using some sort of software. And so there are a variety of different software programs that are available that once you take images of your 2D gels, the software will superimpose those for you to pinpoint which spots are separated between your treatment and control. This can get a bit dicey because it relies upon the methodology and the composition of the gel and composition of the samples being very analogous between the two different two-dimensional gels. So if one of the gels was applied to slightly a different voltage than the other, some sort of other condition differed just a small amount, then you could see variability from gel to gel that quite possibly could cause it to fail in the automated analysis. But assuming the automated analysis works, what will turn up from this is something like this, where we have superposed the treatment and the control using appropriate software. And there are several softwares suitable for this that are specialized for this. So this would be superposition of the treatment and the control 2D gel images that have been stained with silver, Kumasi, or whatever the preferred stain is for the particular experiment that you're doing. And in this particular gel, what's going to be the case is that one of the colors of spots, either blue or orange, will represent your treatment protein. So let's say the treatments are the orange spots. Those represent proteins that are unique or heavily enriched in the treatment gel. Control, represented by the blue signals. So compound proteins that are present only in the control or that are heavily enriched in the control are blue. And then the black signals are proteins that are common to both and at approximately the same intensity in both. So then from there, at this point, the experimenter wouldn't know what the identity of a particular protein of interest is. Like, say, hypothetically, this particular protein that's quite orange and hence heavily enriched in the treatment, we wanted to know what is the identity of that protein, or this protein for that matter, or any of these proteins. How would we go about doing that? Well, from the gel alone, all we can tell is the approximate PI value and the approximate molecular weight. And since if we think about a given cell, there are lots and lots of proteins of a particular molecular weight that are within a cell and a particular expected PI value within a cell. And so this 2D gel alone is not going to give us the identity of the protein. And so that's why this technique is now typically coupled to mass spectrometry, where what would happen next is that these individual signals, these individual proteins, would be cut out of the gel, either using a scalpel in a non-automated fashion, or in some cases with sophisticated equipment, this can be done automated, so the system itself will cut this signal out of a gel. So we excise it or cut it out of the gel for mass spectrometry analysis to give us information on what the identity of that particular protein is, to take this a step further and have understanding of what proteins we are looking at here. So let's say we've excised a protein of interest from the gel. Then at that point, what is typically done is a couple of things. A series of cleanup steps are generally necessary before that protein sample is suitable to be introduced into the mass spectrometer, including removing the gel components from the protein so that the protein is relatively clean and pure to be introduced into the mass spectrometer. Additionally, in some cases, we can collect molecular ion data for a complete intact protein. However, more commonly than that, what is typically done is that the protein is digested using proteases to break the protein into smaller, simpler fragments for analysis by mass spectrometry, rather than trying to evaluate the typical full intact protein, which is often very difficult to do since these are large biomacromolecules. Generally, what happens with these is that they are broken into smaller fragments by enzymatic digestion. So what generally happens is that, to show this schematically, we would have the large protein here, and it is subjected to enzymatic digestion. And sometimes this digestion actually occurs while the protein is still within the gel matrix before the protein is even cleaned up and purified for evaluation by mass spectrometry. So it is digested, and a typical enzyme that is used to digest the protein, to give you an example of this, is the serine protease trypsin, is the most common enzyme used. And what trypsin does is it cleaves the peptide bond at the carboxy side of the amino acids arginine and lysine, two of our basic amino acids. So trypsin is going to cleave or break at the carboxy side of the basic amino acids, arginine and lysine. So hence, if we had a sequence of proteins, which I will just put in using their one-letter abbreviations, and say this was our protein sequence written out using the one-letter abbreviations, remember that we write this out from the N-terminus being on the left, going back to our initial review of biochemistry, C-terminus on the right. That means that toward the right-hand side of each of the individual amino acids here is where you would find the carboxy side. And so what we do is we go through and we look for the one-letter abbreviation for arginine, Arginine is R, so here would be our R amino acid. And so if we were digesting this with trypsin, which cleaves on the carboxy side of arginine, what would happen is that the protease, the trypsin, would cause a breakage in the peptide bond right here on the carboxy side of arginine, breaking this small protein that we have listed the sequence for here at this position, breaking that covalent bond, releasing those two peptide fragments. Lysine has the one-letter abbreviation K, which we don't see in this particular sequence. Remember that L here stands for leucine, not lysine. Lysine is K. We don't see a K here, and so there are no lysine carboxyside termini to cleave here. So as a result of digesting this with trypsin, the product would correspond to two peptide fragments to analyze, corresponding to the sequence that I've listed here on the left, as well as the tetrapeptide sequence that I'm listing on the right here. And you can imagine in a typical protein, there are between 200 and over 1,000 amino acids. And so with several arginine and lysine residues, what happens here is that by digesting with trypsin, we break a massive protein into much more simplified fragments that are much more suitable for mass spectrometry. So after that trypsin digest, generally what happens is that these peptide fragments are purified, and then they're subjected to mass spectrometry, and the mass spectrometry will give the molecular ions, and those molecular ions can be matched with amino acid sequences that correspond to those particular molecular ions. Because given the fact that there are only 20 standard amino acids, there's a limited number of combinations and possibilities of molecular ions that can result here. Because remember that the molecular ion is generally equal to the molecular weight of that fragment plus or minus 1, corresponding to the negative or positive charge that took on in order to be ionizable. And so we can match that up with specific amino acid sequences. Now keep in mind that that is a simplification, because in many cases there are post-translational modifications that occur for proteins, particularly in eukaryotes, where the protein specific amino acids pick up glycosyl groups, meaning carbohydrate groups, or they're phosphorylated, or other types of modifications that would make these molecular ions not quite match up with the exact predicted amino acid sequence. But the general idea here is that information can be gained about enough of these fragments in order to establish what some of the peptide sequence is within that protein. And then that partial peptide sequence can be compared to a database of all of the proteins that are known from that particular organism in order to match up with some protein from that database. So that's how we work around the issue of post-translational modifications in many cases. So just to summarize that a bit in terms of how to actually identify a protein based on these fragments, the fragments that resulted from digestion of the protein with trypsin or whatever particular protease was used there, is that the mass spec is going to give molecular ions, and some of those ions should hopefully be for proteins without post-translational modifications, or specifically peptide sequences that don't have post-translational modifications, because there aren't post-translational modifications throughout every single amino acid, only certain ones of the sequence. So ideally, with some luck, some of those ions will be for peptides that don't have any post-translational modifications, which I'll put in as PTM. And those molecular ions can be matched with sequences from a database of known proteins or predicted proteins from a particular organism. and that will allow the identification of the particular protein of interest. And that identification can be further supported by going back and looking at the PI value that was determined by the 2D gel and the molecular weight approximately of the whole protein that was determined by the gel. Because if one were to look at the database of known and predicted proteins and they saw that, for example, this peptide fragment up here corresponded to a particular protein we'll call protein Z, and then we looked at protein Z and we were like, wait, the PI value that's predicted for that protein and the molecular weight of that protein doesn't match up at all with the molecular weight of this protein that we had digested, then we would need to go back to the drawing board and look at other possible proteins from the organism. But if we look at this particular peptide fragment, we see that it matches a peptide fragment in the database that corresponds to a particular protein Z, and protein Z also has a predicted PI value and a molecular weight that correspond with what we saw on our 2D gel, then we can be pretty confident that we have accurately identified it. So this all sounds pretty good and straightforward on paper, but do be aware that there are some challenges and limitations to this approach. Namely, those challenges and limitations of using 2D electrophoresis and mass spectrometry include the following. So I'm going to call this 2D gel problems, because every experimental technique has some problems. So if we're using a 2D gel with mass spectrometry as our proteomics approach, one common problem with this is that when we are looking at 2D gel electrophoresis, if we have a low abundance protein, it will be obscured by abundant proteins. And keep in mind that the number of genes that are predicted to be expressed in one human cell at any given time is often greater than 10,000. And if you include the different types of post-translational modifications that can occur of those proteins, there can be predicted to be up to 1 million different proteins present. And there's no way possible for current 2D gel technology to separate that many proteins. So when we see a particular signal on a protein gel, it quite possibly is going to be one particularly prevalent, abundant protein and other minor proteins present as well. And so we would, in many cases, be able to see only the abundant protein, and the less abundant proteins would be obscured. So a common problem is that the low abundance proteins could be important in driving a particular biological observation, but those are going to be obscured by higher abundance proteins. And so an important but low abundance protein could be overlooked. Kind of along these same lines, some types of proteins are not separated or resolved at all using this two-dimensional technique. So some types of proteins don't separate well. Particularly if you have a large class of proteins that have the same or very similar PI values and also very similar molecular weights, they won't be able to resolve on a 2D gel, since a 2D gel relies upon differing PIs and molecular weights. So one example of this is membrane proteins are a large and important family of proteins, and many of these will not separate well from one another on a 2D gel. And so hence a person could readily overlook a change in one of those proteins because they aren't separating within that mixture. Another issue in terms of identifying the proteins by mass spectrometry, we talked about how one could digest with trypsin or some other protease and identify what peptide fragments were present based on mass spectrometry, and then compare that to a database of protein sequences to find proteins that had that particular peptide sequence. But unfortunately, the databases have some limitations. The databases typically have the most common proteins, and so if you are exploring a totally novel, unstudied protein, it quite possibly is not present in the database. So the databases, which I'll abbreviate as DB, are limited typically limited to some of the more common proteins. The databases are, of course, continuously improving and expanding, but there's still definitely limitations on that. So just be aware that these are going to be some of the common problems and challenges and pitfalls that one can run into when they're thinking about practical applications of 2D gels with mass spectrometry as a proteomics analysis. This is not a truly global analysis, meaning it is not going to be able to compare every single protein between a treatment and control. Instead, it's going to be biased toward exploring the more abundant proteins, where the low abundance proteins will be overlooked. It's going to focus on proteins that can be readily separated, because ones that don't separate are going to show up as a gigantic blob signal on the gel. And the identification capabilities are not quite yet where they need to be, because the databases of proteins are limited, particularly if you are studying an organism that is not a well-established model organism. You're going to have extreme difficulty finding a database of proteins for that particular uncommon organism. So now that we have an overview of using 2D gel electrophoresis with mass spectrometry for proteomics analysis, we are going to, in the next video, go on to additional strategies that we can use for proteomics analysis and discuss some of the pros and cons of those techniques relative to the 2D gel plus mass spectrometry approach. So stay tuned for that.